Welcome to Mr. Chalk's Revision Tips. In this video, we will look at
action potentials and salutatory conduction. So, an action potential is part of a process that occurs during the firing of a neuron. During an action potential, part of the neuronal membrane opens to allow positively charged ions inside of the cell and negatively charged ions out. This process causes a rapid increase in the positive charge of the nerve fiber. So an action potential is very rapid in change of polarity that moves along the nerve fiber from neuron to neuron. So in order for a neuron to move from a resting potential to an action potential, a short-term electrical charge that allows an electrical signal to be passed from one neuron to another must be achieved. So the level of stimulation that a neuron must receive to reach action potential is known as a threshold of excitation, and until it reaches that threshold, nothing will happen. So propagation is the action of a potential gradient at the axon helixoid that propagates as a wave along the axon. So if we go and look at how this happens, so going down the length of the axon, the action potential is propagated because more voltage-gated sodium ion channels are opened as the depolarization spreads. This spreading occurs because sodium ions enter through the channel and moves along the inside of the cell membrane. So at a resting potential, the concentration of sodium ions outside of the axon membrane is high relative to the inside, whereas that of the potassium ions is high inside the membrane relative to the outside. So the overall concentration of positive ions, however, is greater on the outside, making this positive compared to the inside. So the axon membrane is polarized. So a stimulus causes a sudden influx of sodium ions, hence the reversal of the charge of the axon membrane. This is the action potential, and the membrane has become depolarized. So the localized electrical circuits established by the influx of sodium ions cause the opening of sodium voltage-gated channels a little further along the axon. So the resulting influx of sodium ions in this region causes depolarization. Behind this new region of depolarization, the sodium voltage-gated channels close and the potassium ones open up. So potassium ions begin to leave the axon along their electrochemical gradient. So the action potential is propagated in the same way further along the axon. So the outward movement of potassium ions has continued to the extent that the axon membrane behind the action potential has returned to its original charged state and is said to have become repolarized. Following repolarization, the axon membrane returns to its resting potential in readiness for a new stimulus or a new action potential to come along. So if we look at salutatory conduction, so salutatory conduction is the propagation of an action potential along a myelinated axon from one node of Ranvier to the next node, increasing the conduction velocity of the action potentials. So electrical signals travel faster in axons that are insulated with myelin. So myelin is produced by glial cells that wrap around the axons and help electrical current flow down them. Myelin insulation does not cover the entire axon, so rather there are breaks in the wrapping. So these breaks are called nodes of Ranvier. So the distance between the nodes is between 0.2 and 2 millimeters. So an action potential traveling down the axon kind of jumps from one node to the next. So this is called salutatory conduction, which means to leap. So salutatory conduction is a faster way to travel down the axon rather than traveling in the axon without myelin.
Thanks for watching.